All right, welcome to our third lecture from Chapter 7, which is all about protein function. In this specific lecture, we're going to be discussing the immune response. In our other two lectures, we talked about oxygen storage and transport, as well as muscle contraction, and this is our third one out of three. And again, just like with the other ones, we're going to be focusing on these principles where the diversity of structures that we see in a protein affects how that protein functions, and how the function of a protein depends on both its structure and the way that that structure allows that protein to interact with other molecules and other proteins. So let's get into talking the immune response. Why do we have an immune system? So one of the main functions of our immune system is to prevent infections. And our immune system is going to defend us from a whole variety of different types of invaders that might want to get into our bodies, including bacteria, viruses, fungi, and worms, and other types of parasites. And so all these different invaders are transmitted by all different sorts of routes, right? We can have inhaled invaders or pathogens that might cause a respiratory disease. They can be ingested, which would cause maybe more gastrointestinal distress. You can also get, if you break the skin barrier, you can have pathogens enter directly into your bloodstream through a puncture wound. And so this is why we have thus a variety of defenses to defend against all these different types of invaders and all these different modes of transmission. So here are a few different ideas about what it means for our different types of immune defenses. So some of our immune defenses are non-specific. They help just defend against microbes in general. So these are things like, these are often things like barriers, right? So things like mucus lining your GI tract or your skin. It just prevents any kind of microbe from just trying to get into the cells of your body. There are also specific types of defenses that recognize just one particular microbe or pathogen, or just a couple of different pathogens that are similar to one another. And we're going to talk more about those specific responses in this lecture. And then we can also divide those defenses into two categories by where they're protecting you. So there's external defenses that prevent entry into your body, as well as internal defenses that protect against that pathogen after it's already gotten into your body. So we have this whole variety of defenses to help try to keep us protected no matter what is making us sick or how it got to you. So let's talk a little bit more about the immune response in general. So when we think about the immune response, we often think about two arms of the immune response, the innate and the adaptive. So oftentimes when we have a new infection that our body has never encountered before, the first response to that new infection is often through the innate response, which is a more non-specific response. It uses several different types of immune cells, some of which you might have heard of, such as macrophages, which are generally just sort of patrolling around your body, looking for stuff and cleaning it up. And so in the case of a brand new type of infection, this innate response can start right away and stop that pathogen from multiplying and spreading throughout your body and from you to other people. And that innate response gives your other response, your adaptive response, time to develop. And that adaptive response is a very specific response that attacks one particular microbe. And it also helps you develop resistance to a future infection by creating a memory of what that pathogen looks like so that your body can recognize it for next time. And so the combination of having an innate response as that first line of defense, giving us time to create that adaptive response that more specifically will target the pathogen and help remember it for later, is a sort of general paradigm for what we think of when we think about a lot of immune responses. And so the types of cells that mediate, particularly the adaptive response, are called lymphocytes. And these are specialized white blood cells that we see in the bloodstream, patrolling the bloodstream, as well as patrolling in the lymphatic system. And you see on the right here a diagram of the lymphatic system, which has your spleen, which is your primary lymphoid organ, as well as a number of lymph nodes, which are secondary lymphoid organs, and all the lymph vessels that connect your whole lymphoid system. And this is a place your body has sort of walled off for allowing your lymphocytes to encounter pathogens and start to develop that specialized, specific adaptive immune response. And so ultimately, at this point, once you've had this interaction and you've trained up those lymphocytes, now your body is able to recognize that invader, that pathogen, to launch an attack against it, and then, of course, remember it for the next time that pathogen tries to enter your body. So how does your immune system recognize a microbe or an infected cell? So, like we mentioned before, the pathogens can enter your body in a number of ways, and often that depends on the type of pathogen. So pathogens like bacteria are often extracellular. They're just floating around, for example, in your bloodstream or somewhere else in your body. And so your immune system has to be able to recognize usually proteins that are on the surface of that bacteria. Or in the case of a virus, we talked about how viruses actually enter into your cells in order to live out their life cycle. And so your immune system needs to be able to recognize what an infected one of your own cells looks like in the case of a virus, or what the surface of the microbe would look like in the case of a bacteria, and to then make that specific immune response. And so lymphocytes are the cells that are mediating this, and lymphocytes produce molecules that are able to bind to antigens on the surface, either of a microbe or an infected cell. And an antigen is just defined here. Once again, it's usually a protein, and it's something that is recognized by either an antibody or a T cell receptor. So antibodies are produced by B cells, and those are really what we're going to focus on in this lecture today. But just be aware that there are other types of lymphocytes called T cells, which produce T cell receptors that also play a very important role in recognizing pathogens. And so once an antibody on a particular B cell recognizes an antigen, that will start to stimulate an immune response, which will ultimately, once again, help to fight off that pathogen and to remember it for next time. So let's talk a little bit more about antibodies. So antibodies have this sort of general Y-shaped structure, consisting of two light chains, which we see on the side, they're smaller, and two heavy chains, which are larger. The sort of ends of the Y is a region where the antigen binds, because that's the region of the antibody that recognizes the antigen. And so B cells both produce antibodies on their surface as well as secrete them into the environment. And that allows antibodies to bind to microbes and recognize them, and also to tag them for cleanup, essentially. So we see here in this picture, we have an example of a macrophage that is starting to eat up a microbe that has been recognized by these antibodies that are floating around in the bloodstream. So each antibody that is made pretty much will only recognize one antigen for the most part, or maybe a very small couple of antigens. But every B cell, or every clone of a B cell, is unique, right? So there's this huge variability that's available in this sequence for an antibody. And so that allows your body to make a massive variety of B cells that are able to detect and bind to pretty much any antigen that exists in the world. And it does this through essentially a process of random selection and random generation of different antibodies, which we'll talk about in a little bit. So first, let's talk about the structure of an antibody, because this chapter is all about using the structures of proteins to understand their functions, right? So here are specific isotype of antibody we're looking at is called immunoglobulin G. There are other types of antibodies that you may have heard of, such as immunoglobulin E or immunoglobulin A, but here we're going to focus on immunoglobulin G, or IgG for short. And so there are a number of interesting features on an antibody. So what's shown in yellow here in this diagram is the constant region, and it contains a number of these different domains that we see throughout, and we'll go more into the structure of all these different domains later as we go forward. And those are, again, we have our heavy chain down here, and then on the ends we have our light chains that are, whoops, I just circled not a light chain. We have our light chains here and here. And then on the ends of our antibody, we have these variable regions that are shown in pink on this diagram. And those, again, are the regions that recognize and bind to different antigens. And that's where we see this huge variability in the different amino acid sequence, allowing this variable area to recognize and bind to a massive variety of different pathogens and antigens. Something also important to note here is we see a whole bunch of disulfide bonds that help to hold the structure together. And so this is particularly important for antibodies because we know that these proteins are secreted out from the cell into the environment. And so we want our antibodies to be able to hold their shape when they're floating around in your bloodstream and throughout your body. And so those disulfide bonds help to really maintain the shape of the antibody in a more wide variety of environments. So at this point, let's take a little bit and think about solving this molecular weight question about antibodies. So the light chain of IgG is about 25 kilodaltons in mass, and the heavy chain is about 50 kilodaltons. So this question, I'm asking you to give the approximate molar mass of an IgG molecule when you analyze it by first gel filtration chromatography and then by SDS-PAGE using beta-mercaptoethanol. So please take a moment, pause this video and solve this problem, and we'll go over it in a little bit. All right, so first let's talk about gel filtration chromatography. So the light chain and the heavy chain of the antibody in typical sort of physiological or environmental conditions, that whole antibody is going to be stuck together as one unit, partially because of all those disulfide bonds that we were just looking at. And so when we do gel filtration chromatography, we're going to see the whole antibody going through the column, right? So you will have, and we know that we have two light chains. And each one of those is 25 kilodaltons. So that would give us 50 kilodaltons of light chains. Whoops. We also have two heavy chains in each antibody. Each one of those is about 50 kilodaltons, which would give us 100. And of course, those are attached together. And so we would see a protein coming out of our gel filtration column that looks like it's about 150 kilodaltons in size by adding together that 50 and that 100. It would be very different if we're doing SDS-PAGE using beta-mercaptoethanol, because as we talked about, this particular, when you run a gel like this, we are using denaturing conditions. And so the protein is going to be unfolded, and in particular, the beta-mercaptoethanol is going to break apart all those disulfide bonds. So in that case, we would see separated out light chains and heavy chains. So we would see on our gel one band for the light chain at 25 kilodaltons, and we would see another band for the heavy chain around 50 kilodaltons. And those would be distinct, separate bands. And because, again, of those denaturing conditions, we would not see one larger band at 150 or any kind of combinations of these 25 and 50 kilodaltons for the light and heavy chains. We would just see them all separated out. All right, so let's talk a little bit more about the structure of those domains. So we were talking about how we have those repetitive structures throughout the different parts of an antibody, of an IgG antibody. And that particular structure that we see repeated a lot is called an immunoglobulin fold. And we see that immunoglobulin fold both in the variable domain as well as in the constant domain. So in the constant domain, you can see the structure pretty well here on the left. You have two different anti-parallel beta sheets that are stacked on top of one another, creating what we can call a beta sandwich. And you can see in yellow here, this is a disulfide bond that connects together those two beta sheets. And you can also see on either side of the sandwich the loops that are connecting the strands, the beta strands. So when we think about, and for a constant domain, the primary sequence of this whole structure is fairly constant, hence the name constant domain. In a variable domain, it's a little bit different. So our variable domain still contains our beta sandwich with our two little antiparallel beta sheets connected by loops. But the loops in this case are what we call hypervariable. And this means we have a huge variety of different amino acid sequences contained in those loops. And this makes sense, right, based on what we've learned so far about structure, right? So for beta sheets, we are going to need a fairly specific, maybe sequence of amino acids that's going to be more likely to form and stay stable when it's put into a beta sheet shape. On the other hand, loops, as we know, do not have a constant shape or structure. They're often more mobile. And so to have a lot of more variability of the different potential amino acids at each position within a loop is makes a lot of sense, right? Because our loops do not have to have a constant or particular shape that they're constrained to, and that allows for a pretty big variety in what amino acids you can have there. So this variability in those variable regions allows for, again, antibodies to bind a massive variety of antigens. And the quality of that binding is that it's really specific and it's really strong. And so we see in this diagram, this sort of filled in green molecule here. This is an antibody, and we see those hypervariable loops kind of folded kind of up and around this purple antigen. And we can see that this is a sequence of amino acids. You can see some of the side chains there. You might recognize some of them. So often antigens aren't limited by their size. This is a very small sort of piece of an antigen, and the piece of an antigen that is actually recognized by those hypervariable loops, we call that an epitope. And so if your antigen is a relatively large protein, it might have multiple epitopes on it, where a different antibody might bind different pieces or areas of that antigen. So hopefully that makes sense. An epitope is a small piece of an antigen that is recognized by the antibody. And we'll see another example of that in a little bit. So first of all, let's talk about how we are able to generate this massive diversity of antibodies. And so this occurs essentially through somatic recombination, which is happening during the process of making antibodies. And so you can see here in this little diagram, we have what's called our germline configuration, and this is what's actually in your DNA, in your genome. And so there's a number of different segments of different types. There's V, D, and J segments, as well as constant regions. And then throughout this process of recombination that's happening during transcription of the gene from DNA to RNA before it gets translated into protein, we have splicing out of a bunch of different segments. So ultimately, your antibody will choose, just one V segment, one D segment, and one J segment, and it'll choose randomly from among the variety of them that you have in your genome, as well as choosing one or more of the constant region areas. And then in addition to sort of randomly picking from the V, D, and J that we see over here in green, red, and yellow, when you're splicing them together, we also have a process that sort of adds in random nucleotides in between some of the segments as well. And so this helps to generate even more diversity, because you'll have, this will give you a whole big variety of codons, which turn into a whole big variety of amino acids, which will affect the primary structure of those hypervariable loops. And so we can see like all these different color of those segments that got chosen, and those end up in that hypervariable region. And this example is just showing you in the heavy chain. So we have all these mutations that can occur and that can happen during this recombination process, which happens during transcription, and also when B cells are dividing. So This is happening when you're actually mounting an immune response. You have, this big variety of B cells in your body, and maybe one or two of those B cells will recognize the pathogen that's infecting you. Those B cells are going to get a signal to start dividing and multiplying because you want to make a whole bunch of B cells that recognize the particular pathogen that's in your body. But you don't want to make a whole bunch of every kind of B cell, just the ones you need. So during B cell division, there will also be additional sort of random mutations that occur, and some of those mutations will help the antibody bind better to the pathogen, and those B cells will again get chosen to keep dividing and keep fighting off the pathogen. And you could also get mutations that occur at this point that don't help the B cell recognize the pathogen, and those B cells that don't recognize the pathogen as well will not get a survival signal, and they will eventually die off. And so I also just want to make really clear that this antibody diversity that we see, especially after this VDJ recombination, this is happening long before a pathogen gets into your body. This is just a normal process that's happening while B cells are developing in your body. And so you make this big variety of B cells, and your body goes through this process where it essentially tests them, and it makes sure that none of them recognize your own self. You don't want your B cells to recognize proteins that are part of your own body, because then your immune system will start attacking you. So B cells have to pass this test, and all the B cells that don't recognize you are allowed to live, and then they just hang out waiting for the pathogen that they recognize. Some of them will see their pathogen, and some of them will never see their pathogen. But this can cause problems, right? So if you have an issue where this testing process where you are asking your B cells, do you recognize your own body? And if so, get deleted. If that goes wrong, you can have what we call autoimmune diseases. And this is when our bodies have made B cells that recognize proteins or cells in our own bodies. And so this table here, it shows some examples of antibody-mediated autoimmune diseases. One example that you might be familiar with is this one in the middle, which is insulin-dependent diabetes mellitus. And so this is where you make antibodies that recognize beta cells that are in your pancreas. And ultimately, what happens is your body loses its ability to make insulin, and that affects your whole process of metabolizing the food you intake in your diet. So so here's a number of different other diseases. All of these are mediated by antibodies that recognize specific cells or tissue in your own body, ultimately essentially destroying them and causing a wide variety of symptoms depending on which tissue is affected. So we talked a little bit when we were talking about quantifying proteins about ELISAs, right? And so ELISAs are a type of assay where we use antibodies to recognize our protein of interest. And so you may have wondered, or maybe not, you may not have wondered, how do we get those antibodies that we can use in tests like an ELISA or for other purposes in the lab? So one way we can make antibodies that's relatively straightforward is to actually use an animal as a little antibody factory. And so we can do this by injecting an animal. Often we'll use rabbits. You can also use mice. You can also use other animals. You'll inject them with an antigen. Their body will mount an immune response. And you can then collect lymphocytes, or you can just collect serum, or you can collect other fluid where the antibodies might exist, and you can purify out antibodies from the animal that you asked to make them for you. In this case, you would make what we call polyclonal antibodies. And so these are antibodies that recognize that one antigen, but they'll recognize different regions of that antigen, which we see in this figure down here. And this brings in back that concept of epitopes that we mentioned earlier. So different areas of the antigen have specific amino acid sequences that are exposed on the surface. And so you can imagine you can make different antibodies that recognize different regions of an antigen because there are different amino acids there. And so you'll have different types of interactions with different types of antibodies. And so, and again, these are called different epitopes. And so this polyclonal response means you make many different antibodies that recognize a variety of epitopes. Oh, yeah, I wrote that on the next line, didn't I? Sorry. And so, yeah, and so this is a really relatively quick process, but it's less specific, right? Because you can imagine maybe in different environmental conditions, some epitopes of the antigen might bind with different strengths to different of those polyclonal antibodies, and you might not get maybe a uniform type of binding. And so a lot of time in the lab, rather than using a polyclonal antibody, which can introduce some variability or some error into an experiment, you want to make what we call monoclonal antibodies. And so monoclonal antibodies, let's start with what they are. So those are just one specific antibody. So our example of our antigen again is down here. A monoclonal antibody is just one particular antibody that only recognizes one particular epitope on that antigen, and it arises from a single B cell, right? Because every single B cell that develops recognizes one unique thing. So you'd use one unique B cell as your start, and so that ensures that you have a monoclonal antibody that arises from that single B cell ancestor. And so the way we do this is in the lab, we'll fuse together lymphocytes, specifically usually B cells, with myeloma cells, and to produce what we call hybridoma cells. And so these are cells that, because they're fused with these myeloma cells, these are cancer cells that want to divide indefinitely, and then you'll fuse them with B cells. And so each hybridoma cell has merged with one single B cell, and so it's going to divide for a long time, but it's only going to make one type of antibody. And so this, as you can imagine, is a lot more time consuming to produce than our first method where we just injected an animal with our antigen. But monoclonal antibodies are extremely common uses as in research as well as in biologic drugs. And so you might have heard of monoclonal antibody treatment in the context of SARS-CoV-2, of COVID-19 disease. You also may have heard of them to treat several autoimmune diseases, as well as some cancers. So with that, now you are ready to answer the Chapter 7 immune response practice questions.